Okay, hi everyone. My name is Millie Plotkin, and I am co-chair of the Social Media Committee for the Academy for Eating Disorders. I'm very excited today to be talking to Dr. Cindy Bulick from North Carolina in America and Dr. Nick Martin from Brisbane, Australia. And we're going to be discussing the new results that have recently come out in an article in Nature Genetics on the genetics of anorexia nervosa. So why don't I give you each a moment to talk about yourselves and give an introduction. So Dr. Bulick.
Sure. Thanks for talking with us today, Millie. I'm Cindy Bulick. I was the lead principal investigator of the EDGI study, which was the Anorexia Nervosa Genetics Initiative. I'm also the co-chair of the Eating Disorders Working Group of the Psychiatric Genomics Consortium. And together, those two groups sort of pulled together all of the samples that came out in this Nature Genetics paper.
And I'm Nick Martin. I'm at the Queensland Institute of Medical Research in Brisbane, Australia, and I've led the Australia-New Zealand arm of EDGI down here. I'm here representing the sort of international aspect of the EDGI project. Not on the call are the very large groups from Sweden and Denmark who've made and the UK, I should say, who've made huge contributions to this effort.
Great, thank you both. And I really appreciate you both taking the time to be on here, especially Dr. Martin. I know it's very early in the morning there.
Fine.
So I'm going to start by reading out the title of this study, and then I'm going to ask some questions to break down what do these terms mean and what is it this study has shown. So the title is Genome-Wide Association Study Identifies 8 Risk Loci and Implicates Metapopsychiatric Origins for Anorexia Nervosa. That's kind of a handful. So let's start with Genome-Wide Association Study, also known as GWAS. What does that mean and how is it useful for doing studies on eating disorders?
Sure, I can run with that one, Millie. So it's really the current most widely used and most successful technique in genomic medicine, in psychiatric genomics for sure. And we've gone through this history where we were sort of looking at one gene at a time, which was a little naive in retrospect. And what GWAS allows you to do is actually get a large sample of individuals with a particular illness or trait and a large sample of individuals who presumably don't have that trait. And you basically are slathering like 1 million markers across the genome, and you're comparing the genomes of the people with the illness to the people without the illness. And what you're looking for is the areas in the genome where differences between those two groups arise. So, Nick, anything you want to add to just what a GWAS is, or?
Well, just to make the point that that this only began in 2005. The whole thing's been enabled by this absolutely incredible bit of bioengineering, creating these chips the size of a postage stamp. Well, they used to be actually. They're only about a tenth the size of a postage stamp now, on which you can type up to 1 million gene variants. And when this began in 2005, these chips were $2,000 each. So you couldn't do very big experiments, but now they're down to about $20 each. Still quite expensive, actually. But it means we can do a lot more. It's still quite expensive because the other thing we've found out in those 12 years is you can't do this small. You need huge numbers. So even though the chips are only $20 each, I mean, ideally, we'd be doing this on 100,000 cases and 100,000 controls. So that they do become quite expensive experiments. But the great thing is we know they produce the results, the right results. You know, Cindy and I go some way back in this subject to a time when we were using various techniques that we were very hopeful of but just didn't work. And then finally, we've got a tool that really works and is producing results.
That's exciting. It's amazing how far things have come since you started working on this. And
I'm 69, and everybody says, "Oh, when are you going to retire?" I said, "Why would I retire when the subject is at the most exciting phase of its existence? You know, there's no way I'm getting out."
Right. So the next part of this title is 8 you've identified 8 risk loci. So what are loci?
That's a really important question, and thank you actually for asking it, because it's something that's a little hard to convey, because people always think that we're identifying 8 genes. We're not actually identifying 8 genes. We're identifying 8 regions on the genome that differ between those cases and those controls. And those loci, if you get a chance to look at the figures, they sort of pop up above the significance line and say, hey, it's like shining a torch on the genome and saying, look in these 8 positions for genes that might be relevant to anorexia nervosa. And some of those loci just had a couple relevant genes in them, but one of them, I think, Nick, if I'm correct, had upwards of 30 genes in it. So it's sort of just, it's directing your attention to where on the genome you should be looking for risk genes.
So is it fair to see that these are, rather than individual genes, that they're groupings, sort of families of genes? Would that be a good way to describe them?
I think what it really means, I mean, it is possible that there's more than one signal at each of those loci, but most likely is that there's just one signal there. But in some cases, we're going to have to do quite a lot of extra work to find out which particular one it is, to find out which amongst those 30 hits 30 genes that Cindy talked about, which is the culprit. And that is, there are standard techniques for doing that, but those are really expensive and require laboratory work that sometimes can take years. Fortunately, not all the loci are like that. Some of the loci, there's a screamingly obvious gene right underneath it, one that many cases we often know about from studies of other conditions, other psychiatric conditions, and we can sort of zoom in on that much more quickly. But for some of them, it's going to be quite a lot of work.
Fascinating.
And Millie, I'm just going to take Nick's comment one step further.
Okay, certainly.
It's exciting that we have 8. And that sort of suggests that we're doing the right thing. We're looking in the right places. We should continue with this kind of work. But our expectation is that we're actually probably going to get 100 or more of such loci as our sample size increases. And Nick mentioned that we're going to go up to at least 100,000. But what this does is, these first 8, I kind of say it's both a breakthrough and a starting point at the same time. It's a breakthrough insofar as it's saying, yes, y'all are on the right path. Let's keep doing this. And it's a starting point saying you really do need a lot more samples so that we can identify more loci and more genes. And they started sorting themselves into sort of discernible biological pathways.
Yeah,
okay.
So I mean, it's sort of like a hockey stick curve. When you do this work, if you look at the increasing sample size and then the number of loci that you find, you go along for quite a long way without finding anything. And that was our sort of disappointing experience with what we call ED1 and ED2 papers, where we had samples of 1,000 and 3,000, but essentially we didn't have much at all. And the great thing about this new result is we know we're on the upward part of that curve now. And we can predict that, we can almost predict how many more loci we will find as the sample size increases, just from the shape of that curve. So it's very exciting.
It is. And I can see why with a sample of only 1,000 or a few thousand, it would be really hard to find much. But as you're getting up, and why you want to go towards 100,000.
We say that with hindsight, but that wasn't obvious to us when we did the first study. We were full of hope when we had our first 1,000. We thought, wow, that's a big sample, because it was a big sample compared with the earlier single gene studies that have been done, but it was just nowhere near enough, and neither was 3,000. So, anyway, we know that we're on the right track now.
Good. So I'm going to go into the last part of this title, metabo, I have a hard time saying that, psychiatric. So I know that's referring to the metabolism. So this is, I think, the thing that's really getting a lot of press, is how are the metabolism and psychiatry playing together? What did you find out?
So this really, if people have access to the paper, they can look at figure 2, because that's the one that sort of leads to this phrase, metabopsychiatric. And what we do in that particular analysis is something called genetic correlations. And this is kind of a cool new technique as well. It was developed by my son, if I may have a moment of maternal pride. And what it allows us to do is we can take our anorexia nervosa sample and all the genetic information in that GWAS, not just those 8 loci, but all of the genetic information above a certain threshold. And we can compare that to GWASs that were done on other disorders and traits on completely different samples. And it basically allows us to determine the extent to which the genetic factors are shared between those traits. And I'll give you an example. So at the top of this figure, we see a really strong positive genetic correlation between anorexia nervosa and obsessive compulsive disorder. And in fact, it's one of the strongest genetic correlations we see between pairs of psychiatric disorders. And I think the cool thing about that is that's what we see in the clinic. You know, we commonly see comorbid anorexia nervosa and obsessive compulsive disorder. But this says that one of the reasons behind that frequent co-occurrence is genetic, because there are shared genes across those two conditions. And then we also see those positive genetic correlations with depression, anxiety disorders, and schizophrenia. So that's the second half of the hyphen, the psychiatric part. Nick, you want to add anything to that before we get into the metabo part?
Or just simply, I think as a clinician, you would say that you also find anorexic females with brothers who've got obsessive compulsive, you know, so is sort of further support for this, yes.
Right, that's true.
That's all.
Those two illnesses tend to sort of travel together in families. Right. The other little thing before we get to the metabo piece, because I'm working my way down the figure, is we found this positive genetic correlation with physical activity. And the physical activity GWAS that we used was measured physical activity. So people wore accelerometers on their wrists. This was in UK biobank. So we really knew it was measuring how much activity they were actually doing, not self-report. And so there's a positive genetic correlation between anorexia nervosa and physical activity. Now, what's interesting about that is we know that high physical activity is a very difficult to treat symptom that's part of anorexia nervosa for many people. They pace, they move around a lot, they might go out and exercise and run and bike, even when they're at very low weights. And our explanations for that have generally been psychological. You know, they're trying to work off calories, they're trying to lose weight, maybe a little biology. Occasionally people would say they're trying to warm their hypothermic bodies. But this suggests that they might also be prone genetically to high physical activity. So there might be an underlying shared biological reason that anorexia nervosa and high physical activity go hand in hand.
Yeah, I think that's fascinating. There's certainly been animal research with rat studies and physical activity, but there's always a hesitation of how much do you translate a rat activity to a human activity, and to see that there is some biological basis. I think that is very important.
What about the metabo bit, Cindy?
Yeah, that's next. I'm working my way down the figure now.
Okay, right, okay.
Yeah. So then when you start looking at the negative genetic correlations, it still means that the same genes are involved, the same genetic factors are involved, but they're working in opposite directions. And what we see is we see things like fasting insulin, BMI, weight circumference, type 2 diabetes being negatively genetically correlated with anorexia nervosa. And that whole cluster of findings basically suggests to us that it's not only psychiatric genes that are correlated with anorexia nervosa, but also genes and genetic factors that are associated with metabolic factors. And we're not seeing this magnitude or consistency of metabolic genetic correlations with other psychiatric disorders. There are some, but not as robust as these. And that's what led us into this concept of calling anorexia a metabo-psychiatric disorder.
So I know in a lot of the reporting that I've seen on this study, some of the headlines say there's a correlation with metabolism. And some of them go a step further and say, and I've heard people discuss, oh, this proves that there's a metabolic cause.
Right.
Can you clear up what's true and what's not true there?
Yeah, I think the most important thing is that the people who write titles for articles aren't necessarily the people who write the articles. And interestingly, I think most of the journalists have been really responsible in trying to report this stuff accurately. Proof is not something that we necessarily aim for in this kind of research. If anything, what this is really doing is it's opening up a new way of thinking about the illness. The genetic correlations are that, they're genetic correlations. So that word is right in the analysis. But the important thing is now, as we saw hints of this in our previous smaller study, that have now become more robust in this study. And even more so because we partialed out the variants that are known to affect BMI. So there's still this positive correlation between anorexia and metabolic factors, even when you account for the variants that influence BMI. So we're feeling more confident in this association, but I don't think we would ever go quite so far to say as it proves anything causal. Nick, any arguments there?
No, all quite right.
Good.
Any additions?
Well, I mean, you know, it's just the old, it's like statistics 101 first lecture. You know, correlation doesn't necessarily equal causation. So I guess I'm slightly at odds with Cindy and wondering whether we've done quite enough to explore the direction of causation in some of these relations yet. And that is yet another argument for needing a much bigger sample size, because to some extent we don't quite have the statistical power yet to tease that apart.
Yeah, I mean, we've done all we could given the existing power. As our sample size goes up, there are other techniques that you could use that can really start not only to sort of sort out causality, but also direction of causality. But we're still just slightly underpowered. We really need to have at least 10 loci to be able to feel more confident in that. So we're close, but no cigar yet.
So is there anything else that you've seen in the reporting or in discussions that you've had in the last week that you think are major misunderstandings of this study that you would like to address?
I haven't seen this one, but I have cautioned people against it. And that is ever taking from this kind of research that genes are destiny. You know, that is the absolutely last thing that this research is supposed to convey. You know, I think when we think about an individual person's risk, you've got this sort of like 4 different buckets of risk and protective factors. You've got genetic risk factors, you have genetic buffering factors, you have environmental risk factors, and you have environmental buffering factors. And anybody's individual risk is whatever critical combination of those 4 things that they have. And, you know, in no way are we saying that if you have a certain number of variants, you know, you're destined to develop anorexia nervosa. We're really encouraging people to sort of embrace complexity, to think carefully and creatively about probabilities, and to understand that this is a complex business. You know, like you said before, you know, this isn't proof of anything, but this is a really interesting way of perhaps broadening how we think about this illness and what some of the factors are that contribute to some of the really core perplexing features of anorexia, like the extreme weight loss, like the frequency of losing weight again after being renourished, and that cycle of weight gain and weight loss, weight gain and weight loss. And also that inexplicable hypermetabolic period that people go through when they're being renourished that we really haven't had a good explanation for. You know, what's going on there metabolically when their bodies need 4,000, 5,000, 6,000 calories, and no logical refeeding equation could ever predict that they would need that much to gain weight. You know, so I think we've had these interesting metabolic questions all along, and now we have some suggestion that, yeah, it's really worth spending some time thinking about those metabolic questions more deeply.
Thank you. And definitely thank you for addressing that notion of genetics are destiny. I know as someone who's recovered from an eating disorder myself, when a lot of the genetics discussions first started 15 years ago, it could sometimes feel a little bit hopeless of, well, if this is my genes, you know, what can I do? You know, I'm kind of stuck here.
Yeah. Yeah.
So I think it is important for people to realize that this is to be overcome.
Yeah.
I mean, I get the point I make, you know, it's sort of an analogy is, look, myopia, short sight is completely genetic. So what do? You wear glasses. You know, if you're very short in stature, what do? You go and buy high heels. You know, if you don't like your hair color, you go and buy some hair dye. There are lots of things you can do environmentally to change genetic destiny, if you like. You know, so just because something's genetic doesn't mean there aren't things you can do something about.
Thank you. Those are excellent analogies. I appreciate that.
Yeah.
So the last part is going to be transitioning, I think, a bit into your newer work. So this study was specifically on anorexia nervosa. Do you think that there is anything from this study that could be translated into the other disorders, atypical anorexia, bulimia nervosa, binge eating disorder, or at this point, is it really way too early to be making any of those leaps?
Yeah, I mean, we were able to break down the sample into those with and without binge eating. But again, that cut our power, you know, and we couldn't really draw any clear conclusions that there were differences. And that's where we have to go. This was a start. Anorexia nervosa was step one. And the next adventure, the Eating Disorders Genetics Initiative, or EDGI, is going to broaden out, and it's going to include bulimia nervosa. It's going to include binge eating disorder. And that's going to be fascinating, you know, because one of the things about our field that is so interesting is, yes, we have people who develop anorexia nervosa and that it stays anorexia nervosa for their entire life. We have people with binge eating disorder who have binge eating disorder and they never have another eating disorder. But we have all these people in the middle who like crisscross back and forth across time. And it's going to be really fascinating once we genotype all those people to see if we can understand what that phenomenon is all about, and whether we can predict in the beginning who is going to cross over, who is going to have a complex course, who is at risk for poorer outcome. You know, once we really have these large samples across the eating disorders, we're going to be able to ask a lot of fundamental questions about the differences of genetics within the eating disorder or underneath the eating disorder umbrella.
So Dr. Martin, I know that you wanted to talk a bit about how you were going to be getting these new people in.
Yes, well, I mean, one of the painful things about the anorexia study we did was being contacted by so many patients who say, well, I don't actually have anorexia, but I've got bulimia, can I take part? And unfortunately, we sort of had to say no because we had such limited funds for genotyping. We could only take people who strictly were anorexic. But, well, we're hoping to get funding for EDGI that will enable us to go across all the eating disorders that Cindy mentioned, in particular bulimia and binge eating disorder. And so we were actually, as a result of the publicity for this paper, we were able to scramble to get ethics approval and get our website up for EDGI. So we've already recruited 426 patients as of yesterday morning, you know, who are ready to take part. And then I was on the phone yesterday afternoon to our friend Sarah Maguire, Cindy, who's come up with a little bit of money to help me start getting the saliva samples in from those people, which is the very first stage of the process. So, yeah, we just need some money to do this. And just like we can predict the number of extra genes we'll get with a sample, you know, increase of 1,000 cases. So I can tell you the number of samples we'll get with an extra $1,000 because the spit kits and the postage are all a very fixed cost, and it just absolutely scales with that. Anyway, we're very enthusiastic to do that. One of the things I'd like to emphasize about the success of this effort so far has just been the extent to which it's completely dependent upon volunteerism in response to media, both conventional media, but particularly social media. The demographic, the main demographic for anorexia is young women, and they're precisely the people who use Facebook and Twitter, and we did extremely well in recruiting through those media. And actually, I'd appeal to anyone who is watching this to help that recruitment just by simply retweeting from Cindy's tweet account, the links to EDGI. That could be very helpful to us. And of course, we're recruiting across Europe, across the US, and certainly across Australia and New Zealand.
So that brings up an interesting question for me, or actually two questions. So you mentioned anorexia nervosa primarily affects women, but I know in some earlier discussion that we had that you definitely need more men to join the sample.
Millie, out of our almost 17,000 women, out of our 17,000 participants, we only had 247 men. So
wow, that is a small group.
Below what we would expect, right, based on epidemiology. So once EDGI kicks off, we're going to be pushing. We're really going to be out there and encouraging men to participate. We're going to get some prominent men who have had eating disorders to really help us encourage other men, because they're out there on social media too.
Absolutely.
We'll try to get ourselves in there in the men's magazines and wherever else men's are hanging out, in gyms, and meet them where they're at, And the neat thing about EDGI, it's going to be so much easier to participate because we collected blood for Angie. But again, the technology has advanced so quickly that we can just, spit into a tube now and do the same work that we did with Angie, just with saliva. So it'll be so much easier for people to participate, and hopefully that's going to sort of get men on board as well, and help us understand if the genetics of anorexia, bulimia, and binge eating disorder are the same in men and women, because we can't answer that question yet.
And then another part of that, as far as diversity, was there much racial diversity in your original sample, and do you think that matters?
Right. So this,
as far as the science.
Yeah. The Nature Genetics study was all of European ancestry population. And there are a couple reasons for that. The main one was when we started this study, we actually didn't have great techniques to analyze GWAS samples that were diverse, because there are large genetic differences across ancestries. And you always want to make sure that any differences that you find are actually due to the illness and not due to the fact that your cases and your controls are from different ancestries, right? So for that reason, when we started the study, it was European ancestry only. Now, things have changed. Our analytic techniques have evolved. We have much better ways of analyzing diverse samples, but we need to make sure that we have adequate numbers of people from other ancestry backgrounds so that we can make meaningful comparisons. So in addition to going out there and getting men for EDGI, we're going to go out there and we're really going to try to recruit a more diverse population as well. Because it's really important. We see anorexia, bulimia, and binge eating disorder across all these different ancestral backgrounds, but we have to ask the question and answer the question whether the underlying genetics are the same.
Yeah, I think it's a very interesting question. I was looking at worldwide epidemiology recently, and this certainly exists in African countries. It exists across Asia, and there has not been a lot of research in this population. So it'll be fascinating to see what you can come up with.
No, well, look, I mean, we're acutely aware of our deficiency, both with respect to different ancestries and also with males. The unfortunate, hard-nosed scientific fact is that to make real progress in those groups, we need sample sizes, up there with the ones we've got for Europeans. So it's going to take a big push to get those. I mean, we're very keen to do that, but it's going to take a big push to get there, and any help we can get with that would be great.
Well, hopefully at least some people seeing this video will sign up.
Yeah. Great.
And we'll be sure to add in a graphic at the end of it with the information about EDGI so that people can find out how they can become part of your sample.
Excellent.
Good, good.
So is there anything else you...
They're barking at me, though.
Maybe this is a sign that we do need to wrap up. Is there anything else you'd like to add? Either of you?
I think I've said all I need to.
Points. Millie, thank you for the question.
Yeah.
Okay, thank you very much for your time. I really appreciate it from both of you. And I hope you have a good rest of the day.
Okay, thanks very much. Nice to talk to you. Bye.